Hello students, welcome to my biology class. Today's discussion is Chromosome Theory of Inheritance. I suppose all of you have listened to my previous video lessons, and in continuation of principles of inheritance and variation, we'll be discussing now the Chromosome Theory of Inheritance. Now, what are we going to learn in this discussion? In our previous discussions, we learned the work of Mendel, monohybrid cross, dihybrid cross, and in another video, I discussed different deviations from Mendelian ratios. We had a short discussion on why Mendel was not recognized during his time. Today we will start with that part and then we'll reach to our destination. We will be able to establish the relationship between the gene and chromosome. You will know who are the contributors in formulating the chromosome theory of inheritance. You will know about the theory, that is chromosome theory of inheritance. You will be able to understand the behavior of chromosomes during movement into offsprings or daughter cells. That means the behavior of the chromosome, how it moves from the parent cell to the daughter cells. Now, we'll start with looking back, that is, Mendel's effort did not go in vain, but for a certain period of time it was unrecognized. But in 1900 and thereafter, it is Mendel's work on the basis of which genetics is based on. Mendel presented his paper, Experiments on Plant Hybridization, at two meetings of the Natural History Society of Brno in Moravia. It was on the 8th February and 8th March of 1865. Now, this was reported in the local newspapers, though it was ignored by the scientific community. Now, since it was reported in the local newspaper, it was not widely publicized because you can just realize in 1865 the communication system was not so well developed as it is today. So Mendel's work could not be known by people on the other side of the world. Mendel's concept of genes, today it is known as gene, but according to Mendel, his terminology was factors. Later the term factor was replaced by genes by Johansen. Now Mendel's factors, he considered them as stable and discrete units. And these factors control the expression of the traits. And above all, the pair of alleles did not blend with each other. Now this concept could not be accepted by the contemporary scientists, which could not explain why continuous variation was occurring in the nature. Above all, Mendel's use of mathematical interpretations, and that too in biological phenomena, was a totally new concept and which was not accepted by the then scientists. They couldn't understand what could be the practical application of statistical analysis in biological phenomena. Though Mendel's work suggested that the factors were discrete units, but it was not possible for him to give a physical proof of the existence of these factors. Neither its chemical makeup nor its physical makeup he could put forward. There were many other reasons why Mendel's work went unrecognized. But after a long time, in 1900, his work was recognized and we'll see how. Mendelism was rediscovered in 1900 by three scientists who worked independently and got the same results. It was DeVries, Correns, and Tschermak. You'll see here, these are those three important scientists whose work brought again Mendel to light. And Mendel was then known as the father of genetics. It was the effort of these three scientists: Carl Erik Correns, a German botanist and geneticist; Hugo Maria de Vries, a Dutch botanist, and he was best known for his mutation theory. We'll learn in our future sessions. It was Erich Tschermak, an Austrian agronomist. He developed disease-resistant crop plants and certain hybrids also. So they were the rediscoverers of Mendelism. Now, during that time, it's after 1900, there was a lot of advancement in microscopy. During the time of Mendel, that the factors had some physical existence and how it looked like could not be explained because lack of advancement in microscopy. Thereafter, with these microscopes, the then advanced microscope, it was possible to observe cell division and also understand how the chromosomes behaved during the different stages of cell division. Now, this advancement in microscopy led to the discovery of structures in the nucleus. Not only nucleus, but also the materials inside the nucleus could be visible, and it was possible to understand what happens in the nucleus just before division or just before the cell division. Later on, these thread-like structures were called as chromosome. Chroma means colored, soma means body. So these are colored bodies because such name was given because chromosomes need to be colored, that is stained for proper visualizing. In brief, let us have a discussion on the journey to chromosome theory, how the chromosome theory was established. Now just think about 1860s when Mendel and Darwin they started exploring to understand how the heredity occurs or what is the mechanism of heredity. Later on, Sutton, Boveri, Fleming and many others they contributed in the discovery of the chromosome, their structure and their behavior during cell division. So before 90s, it's 1860s and then up to 1900, since the nucleus, the inner structure of the nucleus and the chromosome and also the characters of chromosome was not known. So it was not possible for Mendel to explain the nature of the factors. However, after knowing and establishing the chromosomes, it was possible to come to the conclusion to relate chromosome, genes, and the factors. Now, T.H. Morgan, his work gave a concrete proof to relate inheritance of traits and behavior of chromosome and thus finalize the chromosome theory of inheritance, which means that the chromosome theory of inheritance is not the product of a single scientist. Several scientists contributed in different forms, and the ultimate result or the product was the chromosome theory of inheritance. These three scientists played leading role in the chromosome theory of inheritance. That is Walter Sutton, Theodore Boveri, and Thomas Hunt Morgan. this is and this. Now let us see the meiosis and the behavior of chromosomes, because this is the basis for which, or basis on which, the chromosome theory lies. The chromosome was established. The movement of chromosome from cell to cell was understood. It was then correlated with the Mendelian concept of inheritance or transmission of the traits from one generation to the next. So, in 1902, the chromosome movement during meiosis had been worked out. So, this was an important event. Sutton and Boveri noted that the behavior of chromosome was parallel to the behavior of genes. Now, this is very important. That was the behavior of chromosomal movement from cell to cell and the behavior of the factors explained by Mendel. They are collateral. They are the same. Now, let us compare between these two. Let's start with either chromosomes or the factors, whatsoever it is. You just have to find out now. These occurs in pairs. This segregate at the time of gamete formation, and that only one of each pair is transmitted to a gamete. This is very clear. Independent pairs segregate independent of each other. Now, if I take the other part, the genes, they also occur in pair. They also segregate during gamete formation, and only one of each pair is transmitted to a gamete. One pair segregates independent of the other. So if you remember, if I consider this, if you remember the laws of Mendel's laws, law of segregation, which states that for every pair or every pair of chromosome factors or the alleles during gamete formation, they will separate. For example, capital T or capital T small t, whatever be the alleles, when they are in pairs during gamete formation, the two T's will separate into two different gametes. Same was observed with the chromosomes. The diploid set of chromosome during gamete formation becomes haploid set. That means from each pair of homologous chromosome, one complement goes to one cell, another complement goes to another cell. So here we can find that in either of the cases, be it the movement of the factors or the genes, be it the movement of the chromosomes, both have the same behavior. Now, let us try to recall our concept of meiosis. This diagrammatic representation will, of course, help you to understand. See, this is a cell. In this cell, there are 4 chromosomes. They are differently colored and different size so that you understand it. What happens here? You can see here one long yellow, one long orange, one small green, one small red. These are the 4 chromosomes. Now this cell is going to undergo meiotic division. You know, in meiotic division there are 2 stages. One is meiosis 1 and one is meiosis 2. In the meiosis 1 stage, there is reduction division. Meiosis 2, it is equational division. Now let us see what happens, what reduction occurs. As the cell enters into meiosis 1, that means now before entering into meiosis 1, it is in the cell cycle G1, S, G2 and then M. It is in the cell cycle stage. In G2, there is synthesis, DNA synthesis. So every chromosome now has 2 chromatids. You can see here the chromosome has 2 chromatids. Why? Because here DNA synthesis has taken place. This chromosome has 2 chromatids and 2 chromatids. So every chromosome has 2 chromatids. The number of chromosomes does not increase, only the DNA content doubles. Now this cell enters into the meiotic 1 phase or during anaphase stage. What happens? During anaphase stage, the chromosomes are pulled towards the poles. You can see them lying in the equatorial plate from each bivalent. You can see here this chromosome, this chromosome. This is one pair of chromosome. This is another pair of chromosome. So this chromosome, this yellow one and the red one, here it goes to this pole. Orange one and the green one goes to this pole. So at the end of meiosis 1, we have two cells. In two cells we have one chromosome. We have two chromosomes each. Initially we had four chromosomes in the cell. Now at the end of meiosis 1, we have two chromosomes in each cell. It is a reduction division. One is long chromosome and one is short chromosome. In size here what we see. Now let us see what happens during anaphase 2. Now these two, yellow and red, goes to one cell, orange and green goes to another cell. Anaphase 2 occurs in both the cells. In this cell what happens? Here in anaphase 2, the most important part is that is the division of the centromere. So this chromatid, one yellow chromatid goes to this pole, another yellow chromatid goes to this pole. One red chromatid goes to this pole, one red chromatid goes to this pole. Similarly, it happens in the case of orange and green also. So one red orange chromatid and one green chromatid goes to this pole and the other goes to this pole. This results in anaphase 2. Then occurs telophase, means the cell now divides. So this now becomes two cells like this. This cell now becomes two cells like this. So now ultimately what happens? Every cell has two chromosomes, two chromosomes, two chromosomes, and two chromosomes. So the gametes are produced like this. This knowledge is required to understand the similarity between the movement of the chromosomes and movement of the genes. At that time, it was not known that the genes are located on these chromosomes. That was confirmed by TH Morgan, which we'll study later. Now let us study what happens in anaphase. Let us try to understand this part. This is also important. Now, in meiosis 1, anaphase, just now we have understood this topic. That during meiosis, these two chromosomes will go to one pole, these two chromosomes will go to one pole. This, I don't think it needs more elaboration. You can see the stages, what is happening. So one cell has these two chromosomes. Similarly, the other cell has these two chromosomes. It's very clear. There is no problem in this. In the other part, possibility, it is since the chromosomes are also independent to move. It is not necessary that the orange will always go with the green. It may so happen that the orange and the red goes to this pole and the yellow and the green goes to this pole. It's a question of probability. And this occurs because they are independent of each other. That is what in the law of independent assortment Mendel had observed. That is in the dihybrid crosses. When two different characters are studied, they showed independent assortment like what is going to happen now. See what happens. Accordingly, here during anaphase 1, this orange and red goes to this pole and the see here what happens. And similarly, green and yellow goes to other pole. So these are the probabilities because each chromosome they are free to move. So such combinations are possible. The same thing happens in the chromosome and in the factors, Mendelian factors. Now, let us see what happens in the next stage. This is what had happened already. These two already you have obtained. Now in the next meiosis 2, anaphase 2, what happens? There again will be separation of the chromatids. Separation of the chromatids. So as the chromatid separates, then one chromatid, that is the orange one, goes with the green. Here also the same. Yellow goes with the red. So such will happen now. So this produces such type of combinations. I think it's clear now. These two goes into two different cells. So these four types of gametes can be produced. I think it is clear. Now in the other part was not the same combination. Whatever be the combination, that was orange was paired with orange and red, and green and yellow. Now same thing happens. Here there is again centrometic division, and after meiosis 2, the daughter cells are produced. So here you will see, according to the movement of the chromosomes in the meiotic 1 division, we get the combination of chromosomes in the germ cells. Here you see it is orange and green, but here it is yellow and green. Here it is red and yellow, here it is red and orange. That means that the chromosome movements, they are independent of each other, which actually they wanted to establish. So they could correlate the findings, the chromosomal movement, with the finding of Mendel. This is the actually chromosome basis of inheritance. So Sutton and Boveri argued that pairing and separation of a pair of chromosomes would lead to segregation of a pair of factors as they carried. Naturally, because the chromosomes, they are bearing their factors, so as the chromosome moves, the factor also moves. So Sutton united the knowledge of chromosomal segregation with Mendelian principle and finally called it as chromosomal theory of inheritance. So now let us wind up. In a nutshell, what do we understand? The chromosome occurs in pairs, like the factors. They were also in pairs. As homologous chromosomes separate during gamete formation, so do the alleles segregate during gamete formation. I think there is no doubt about this right now. Offspring of the daughter cells receives one chromosomal complement from father and one from the mother respectively for every pair of homologous chromosomes. Now, whatever chromosomes we have, chromosome number 1, there are two such chromosomes, homologous pair of chromosome, of which one chromosome of the homologue, one homologue of the pair is from the father and the other homologue is from the mother. This is the concept. Likewise for all the 23 pairs in human beings. Reduced number of chromosomes restores the paired chromosome after random fertilization. Now you see, this is very interesting. In meiosis, the diploid cell becomes haploid in the gametes. So original cell was a diploid cell, and the gametes, by reduction division, by reducing the chromosome number to half, is the gamete. Now, when two gametes fertilize, is a random fertilization. When two gametes fertilize, then the zygote is produced, and this zygote is diploid cell. So the diploid condition is restored after random fertilization. This is what independent assortment also tried to say. Mendel explained the same thing through his law of independent assortment. And finally, what Mendel did not say at that time, here in this theory says that these genes or the factors are located on the chromosomes. Now after this, the path of genetics opened, lot of work is being done and is being continued. Immediately after so, Bateson, he became a dedicated defender of Mendelism and Mendelian genetics. He coined and popularized many of the terms now used by genetics, like genetics, allelomorph, zygote, homozygote, heterozygote. Danish botanist Johansen, he introduced the term gene to replace older terms, factors, and characters, and he termed phenotype and genotype. So thereafter, genetics took its own pace, and till now it is continuing. Later on in 1902, Bateson published Mendel's Principles of Heredity. So with this, we are coming to an end of this discussion. Genetics has a long way to go. Our discussion will continue in several more video sessions, and I'd like all of you to go through this video lesson once more and try to understand each and every item which I have discussed, because in the coming sessions we'll be having more discussions, and every discussion requires prior knowledge. So don't jump into any other videos right now. Be with us and be in a continuous mode. Don't step jumps, rather maintain a continuity. So I'll see you again in our next discussion. A very, very, very important discussion. We'll have a new concept of linkage and crossing over under the same chapter, and we'll see how genetics is getting wider and wider. Thank you very much for being with me.